Day, and we welcome you on behalf of the College of Pharmacy and its Honorable Dean, Dr. Abdul Majid Al-Qasumi. We will start with two of the talks in this morning session. The first speaker is Dr. Ahmed Abdul Latif. He has a PhD from University Regensburg, Germany, under Professor Achim Gofresh at the Institute of Pharmaceutical Technology. He has a wide experience in nanoparticles and drug delivery. This is basically his specialization. He has worked as assistant professor at the Regensburg University, and later he joined Qasim University. His today's talk is about the nanoparticles delivery for cancer treatment. The title is, as you see, is Active Targeted Nanoparticles as a Smart Way for Cancer Treatment. The time of the talk is 20 minutes, followed by 5 minutes of question answer. So I'll request Dr. Abdul Latif to start his talk. Thank you.
Thank you, Professor Dr. Riyad, for this kind presentation. Ladies and gentlemen, thank you for attention. The title of my talk is Active Targeted Nanoparticles. The title of my talk is Active Targeted Nanoparticles as a Smart Way for Treatment of Cancer. As we know, cancer is a very dangerous disease, and one of the most important treatment for cancer is the chemotherapeutic agent. But the chemotherapeutic agent has many side effects. It cannot differentiate between the healthy cell and normal cell. It destroys all the cells. So, and if we look here for the target, for the distribution of cancer cell, of a chemotherapeutic agent, we will find distribution in every cells in the body. There is a need, of course, to target a specific site in the body just to concentrate the drug and, of course, to treat a specific kind of cancer. Of course, we ask ourselves how we can target the cancer cell. We sang it many times, and we decided to use the active targeted to nanoparticles. I mean, we want just to concentrate the drugs around the kind of receptor. We just used here transfer against the concentration gradient. There is no passive diffusion. We asked ourselves how we can use, which kind of receptor we can use. We choose a somatostatin receptor. Why somatostatin receptor? Somatostatin receptor is the majority of cancer cells express the somatostatin receptor, as published in 1997 by Battle. And we tried to confirm also this result by test all the cancer cells that we have. We try to confirm if all the cancer cells express the receptor or not. And this was the first work that worked how we can determine the expression of the somatostatin receptor in the cancer cells. This is published in Physiology and Biochemistry. We modified and we used a model octreotide, which is a somatostatin analog. The octreotide fits octreotide. And of course, we use the traditional method, for example, the RT-PCR, for example, the using of a specific antibody for this cancer. And the octreotide was labeled, as we see here, with fits octreotide, specifically at the N-terminus phenylalanine, because the amino lysine is essential for the activity. Then the fits octreotide were incubated with different cell line. Before the incubation, we purified these octreotide fits, and of course we selected just the mono-labeled fits octreotide, and using the easy mass, we confirmed this binding by increasing the size to around 389. This is the corresponding molecular weight of fits, and of course the multi-TOF confirmed this binding by increasing the molecular weight also to 1,429. Then we incubated the nanoparticle, or the octreotide fits with different cell line. This is just some example. This is a HeLa cell and fits and MCF7 L929, and this is bone one cell The bone one cell is the positive expression of somatostatin receptor. U89 or 87, this is a negative expression of somatostatin receptor. What we found, and after incubation for just 30 minutes, we found a higher expression and higher shift, as we see here. Sorry for this pointer. As we see here, this is a higher shift using the FACS for the expression of these receptors with HeLa cells and MCF7 and L929. We confirmed this result also by a quantitative real-time PCR. As we found here, what we found by PCR, we found a 30-fold excess of the expression of receptors in compared with the positive and negative control cell. Then we formulated our nanoparticles for this targeting. We started by gold nanoparticles and coating of these nanoparticles by somatostatin agonist. We started this formulation by synthesis of gold nanoparticle citrate using citrate reduction method, and then the citrate were coated with mercaptodecanoic acid and then coated with somatostatin itself. And of course, we confirm it if there is the coating already done or not, using, for example, the zeta sizer. As we found here, there's increase in size from the gold nanoparticles or the unmodified gold nanoparticles and gold nanoparticle mercaptodecanoic acid and the gold nanoparticles after somatostatin coating. Also, the zeta potential went from negative charge to positive charge due to the deposition of somatostatin itself on the surface. Also, we found a redshift in surface resonance, surface plasmon resonance due to the deposition of somatostatin on the surface, as we see here. We found also non-migrated band of the somatostatin gold nanoparticles due to the increase in size of these nanoparticles in compared with the other kind of nanoparticles. These kind of nanoparticles were incubated with HCC 18069 cells. The incubation give us indication that the nanoparticles internalized due to, the nanoparticles internalized due to the presence of a receptor, as the somatostatin gold nanoparticles were internalized in high concentration and compared with the somatostatin receptor blocked by the antagonist. Then, and this was the problem for the gold nanoparticles. The gold nanoparticles is a quenching agent, so you cannot identify the gold nanoparticle inside the cell so easily. So we replace these gold nanoparticles with a quantum dot nanoparticles. This is a fluorescence fluorescence quantum dot, the fluorescence nanoparticles. And also due to the instability of somatostatin, the somatostatin were replaced with octreotide. Then we published the second work for us, the coating of quantum dot nanoparticles with the octreotide. The quantum dot nanoparticles were succinylated using succinic anhydride, and then finally we coated with the mercapto with the octreotide, as we see in this reaction, and then we modified we purified the quantum dot octreotide using the size exclusion chromatography, and of course we measured also the zeta potential and showing the inversion from negative charge to positive charge of the quantum dot octreotide. Then also we confirmed this binding by catching the amide bond of gold of the quantum dot octreotide in compared with the physical mixture, which showed no expression of the amide bond between the octreotide and the quantum dot. The nanoparticles were incubated with different cell line, and this is just example with bone one cell. Bone one cell is a pancreatic cell line, and this bone one cell express also the receptor. The bone one cell showed a higher binding, as we see here, with quantum dot octreotide, and after blocking the receptors, the internalization will decrease, and this kind of nanoparticles showed no non-specific binding with the cells due to the non-expression of due to the unmodification of the quantum dot with octreotide. Um, this built just to express, as we have higher expression of the receptor, that means we have a higher internalization of the nanoparticles. And lower expression of the receptors means, I'll show it in non-internalization for the nanoparticles. For example, this is a HeLa cell which showed a higher internalization of the receptors, and this is the bone one cell as determined by RT-PCR. When the nanoparticles incubated with these two cells, HeLa cells and bone one cell, the internalization with HeLa cell showed a higher internalization in compared with bone one cell. This internalization was around 30-fold excess internalization of the nanoparticles. These nanoparticles were injected now in mice, and of course we tried to know if we have targeting for these nanoparticles or not. What we found exactly, we found a 55% of the quantum dot octreotide circulated and has a massive accumulation in the blood in compared with the other nanoparticles, for example, quantum dot cyclo-RGDF and the unmodified one. So this is the red one, this is the quantum dot octreotide, which showed a higher internalization in the blood. The other particles completely metabolize in liver here, as we see these columns. And this is just transition section from for the unmodified quantum dot and the quantum dot from the liver, just to confirm that there is no nanoparticles of the quantum dot octreotide in the liver. Then we modified our particles by replacing the quantum dot, replacing the octreotide with evaporotide. Evaporotide is more specific for the receptor. So if we have a higher expression and higher specificity, that means we have also a higher internalization. So the quantum dot were coated with evaporotide. Of course, this is the reaction of the evaporotide octreotide using the sulfur SMCC, and then the particles, of course, were identified for the growing in size from around 500 nanometer to around 600 nanometers, and then the quantum dot amino big compared with the quantum dot evaporotide were characterized also using the zeta potential to confirm that the inversion in the zeta potential from negative to positive. The nanoparticles were incubated with MCF7, and we have also the same results, higher internalization due to the incubation of quantum dot evaporotide compared with other one. Again, the flow cytometry confirmed also this binding as this internalization. And of course, we could determine the amount of free peptides that can block the receptor here. It was here around 3.5 micromolar, which can block completely the internalization of this nanoparticle. And this is the most important thing now in our research. Now we have around 75% targeting for the quantum dot evaporotide due to the specificity of the evaporotide to the receptor in compared with the octreotide. This result confirmed that we have, if we have a receptor and the receptor with a higher expression, and of course ligand with high specific affinity to the receptor, that means we can target the receptors or the cancer cell which express these receptors. We modified now this work from quantum dot and gold nanoparticles by mixing both, as we published here in Drug Development and Industrial Pharmacy. This is a mix between quantum dot and gold nanoparticles. The quantum dot has a fluorescence, and this is the most important thing in this advantage, and this is the most advantage of the quantum dot. The gold nanoparticles is easily to be modified and compared with the quantum dot. Now, if we have both quantum dot coated with gold, that means we have a fluorescent nanoparticles, and of course we have surface can be easily modified. And then the quantum dot gold nanoparticles were coated also with somatostatin to show if these nanoparticles have also the same internalization or not, or at least have the same specificity or receptor or not. We confirmed that these nanoparticles uniform with regular size. There is no aggregation And of course, there is inversion from negative charge to positive charge due to the deposition of somatostatin on the surface. And of course, we have red shift due to in the surface plasma resonance due to the presence of gold on the surface. And the fluorescence, still we have the fluorescence of these nanoparticles. That means the gold could not quench the gold nano, the fluorescence of quantum dot in compared with other one. The particles were incubated with different cell line. The particles still have also the internalization, still have the specificity of the receptors, and can be used for next targeting. And finally, also the flow cytometry confirmed this internalization by showing a higher internalization of these nanoparticles. In conclusion, we have now 3 different nanoparticles: gold nanoparticles, quantum dot nanoparticles, and quantum dot coated with gold nanoparticles. All of them can be a carrier, or can be using as a carrier for delivery of cancer. Even these kind of nanoparticles can carry, for example, a gene for antisense therapy, can carry, for example, some kind of chemotherapeutic agent, can target as itself to the nanoparticles, to the cancer cell, and make some modification on in these nanoparticles. I would like to thank all the co-authors in my work. Thank you very much, and thank you for attention.
Thank you, Dr. Ahmed. The talk is open for questions. I think ladies first for the questions. This side, please. Anybody has? Yeah, okay. Give the mic there, please.
Assalamu alaikum. I'm Dr. Amal, pathologist from Uneza College of Pharmacy. First, I would like to thank you very much for this nice presentation. It was really good work and nice work. And you know, we pathologists are not in good relationship with nanotechnology because it may replace us in some areas in diagnostic work. But I have a problem with nanotechnology in chemotherapy exactly. In the area of
carrying,
withdrawals. Withdrawals. Okay. I have a problem in the area of complications and side effects that can happen in vivo when you are using nanotechnology in vivo, not in vitro. There's a lot of studies made in vivo and no problem, in vitro and no problem, but the problem will start when you are using this in vivo. First, from the area of accumulation of the nanoparticles in the liver and the toxicity, here you are trying to overcome the toxicity of the drugs, of the chemotherapeutic drugs, but the problem will be occurring from the toxicity of the nanoparticles itself when accumulated in the liver and when it start to make oxidative stress after it already phagocytes by the phagocyte cells. This is number one. Number two, the excretion of nanoparticles itself. How is going to be excreted? Till now, I think there is no enough studies in this part. The third part also in allergic reactions which can occur due to administration of nanoparticles. Also, the last area can be when you are using the magnetic mechanism in order to direct the nanoparticles. I think this cannot be overcome the blood flow. The fourth problem also, I think, in bypassing the blood-brain barrier. I think all this cannot be overcome easily in vivo. So
Okay.
what's your comment on this?
Yeah,
Thank you
okay.
very much.
Okay,
Thank you very much
you.
for your question. This is a very nice question. This is what happened exactly with me in the beginning. We started this work by gold nanoparticles, which has a size of around 80 nanometer to around 20, 24 or 22 nanometer. And we had this problem in the beginning. The particles were completely metabolized and engulfed by reticular endothelial cells. Okay, because it will be happen usually corona or plasma corona around this nanoparticle will be completely engulfed and completely metabolized in the liver or exceeded by liver. We used here, and this is, I'm sorry for this misunderstanding about the difference between these nanoparticles, quantum dot and gold nanoparticles. The quantum dot nanoparticles was a quantum dot with a nanoparticles and big dilated using a big, with a big chain and a larger particle size. And these nanoparticles have no non-specific binding because and due to the size of these nanoparticles cannot engulf it by the reticular endothelial cells. So if you want to make in vivo or to make the for next work for you, you should start with nanoparticles with larger size and of course should be begilated. Don't use it as it is. Should be begilated and should you take care from the engulfing by reticular endothelial cells. Sorry for, can we?
Okay, thank you, thank you. I think it's done, or you still need to express more?
Okay.
Okay, next question from male side, please. Anybody else?
Okay, thanks.
Okay, thank you, Dr. Ahmed. Okay. Yeah, actually, nanoparticle toxicity is still being worked out. The internalization and metabolism is not clear at the quantum scale. So that's giving a lot of, query, and people are working on that. Anyway, thank you, Dr. Ahmed, for your nice talk and the answer. Gentlemen and ladies and gentlemen, our next speaker is Dr. Abdul Latif Bozoui. He hails from the Al-Qura University in Mecca. He is a hardcore geneticist, and he is from the medical faculty. He has a PhD from John Wolfgang Goethe University in Frankfurt, Germany. And he has a vast experience in the genetics, in the drug deliveries, in the genetic aspects and gene expression, gene delivery. Today he will be presenting something on the antisense peptide, sense and antisense peptide sequences. This is a very interesting field, and the drug development and other areas with the antisense peptides have taken again root. I think 20 years back it was a hard field, and it still came back into the arena. So today's title of his talk is Antisense Therapy, a New Concept to Disease Management. Dr. Abdul Latif, please. Thank you. Yes, excuse me. You have a 20 minutes time, and please adhere to the time, and 5 minutes for the questions.
Okay, I will just start the presentation.
Yeah. Where is your presentation?
Where is it?
You can close it from here. Close it, no problem.
It is. F5. Good. First of all, thank you for inviting me to give this presentation. Some person they will ask what has genetic with pharmacy, but at the moment I think there is no border between the science. Everyone supports the other. So at the beginning I will show what is the title is Antisense Therapy as New Concept in Disease Management. What is antisense oligonucleotide? The oligosense nucleotide, they are short segment of single-strand DNA or RNA They are about 15 to 20 nucleotide, and they are complementary to the target's mRNA. What's mean complementary? That means they can bind to the mRNA. Binding of the sense or antisense oligonucleotide to the mRNA prevent the synthesis of the protein. And to could understand this, I have to explain how the synthesis of the protein starts. So at the beginning, we have the information in the DNA. After the transcription, so the transformation will be copied to the mRNA, and the antisense will bind to the mRNA and cause degradation of the mRNA, which degrade or inhibit the production of the protein. So for the normal traditional drug, the target is the protein, but not the mRNA. So antisense oligonucleotides are now a powerful weapon for the management of different diseases and for the study of gene function. I will give an introduction about the antisense oligonucleotide. It was first formulated in Russia by Grineva et al. 1967. In 1974, Lessinger et al. Introduced the phosphate coupling approach. This phosphate coupling approach is very important reaction for the synthesis of the oligonucleotide. 1977, Patterson et al. Published the first research showing that gene expression can be modified using a single strand DNA to inhibit the translation and protein production. 1978, Samnick and Stephenson used the specific 30 nucleotide to inhibit Rose sarcoma virus in chicken embryos. And in the same year, the same group improved the oligonucleotide by adding modification at 3' and 5' of the oligonucleotide, which inhibits the degradation of the oligonucleotide. In 1979, Dennis Keller showing that RNase H cleave the RNA strand in a DNA RNA specific manner. That means the degradation of the mRNA is not anywhere, but it must be in one site which is specific. 1983, evidence for the existence of naturally occurring antisense RNA in the human genome. That means this function is already established in the human genome, and the body also uses the same strategy to regulate the expression of the genes. In 1998, Fire showed that the double-strand RNA is responsible for down-regulation of target gene in Chemonopsis elegans. Elegans is warm, and they show that double-strand RNA also could inhibit and regulate the gene expression. In 2006, Andy Fire and Craig Milo won the Nobel Prize because of their work on the RNA interference. Mechanism of the antisense oligos, I want to explain so everyone can understand how it works. There is 3 mechanisms for the single-strand oligonucleotide. The first mechanism is the antisense oligos bind to the mRNA, and the RNase H cleave the mRNA in the DNA RNA binding position. The second mechanism is the blocking of the translation. So because the antisense oligos bind in the mRNA, so the ribosome they cannot conduct the translation, and this will inhibit the protein production. The third mechanism, which is the most new mechanism, in this mechanism, the researcher show that using using antibodies or the antisense oligos can bind to specific exon, okay, which produce the skipping of the splicing of these exon. So what will happen after binding of the oligonucleotide? So during the splicing process, the splicer pass the exon 51 and splice exon 48 and exon 52 together. This is in one disease which is named DMD, or Dickinson's dystrophy, muscular dystrophy. Another mechanism is for the siRNA or RNA. This is same, but they have different name. Small interfering RNA or RNA interfering. So how is the principle? The double-strand RNA will be degraded by dicer. After the degradation, we have saRNA. There is another mechanism. We have sequence from RNA which bind together, and after digestion, it will form siRNA. But the second process we found only in the plasmid, retrovirus, or adenovirus strategies. After binding, after the production of siRNA, so the dicer bind to the small interfering RNA and make the antisense free from the sense strand. The next step is the RNA inducible complex bring or bind to the mRNA and causing degradation. At the moment, the most used antisense oligo is the RNA, because the effect of the RNA compared to the single RNA is lower. The double strand or small interfering RNA, the effect is 100 times more higher than the single strand oligos. Advantage and challenge of the antisense oligomer. So we have different potential or advantage. First, the oligonucleotide might be manufactured quickly, so the production of antisense oligos now is much easier. You can order in 2 or 3 days and you get it the next day. The potential of sensitivity to therapy can be measured easier, so you can see if there is effect by using real-time PCR or any Western blot or anything. So the next potential is to produce longer lasting response. So the effect, you can see it for a long time. And the potential for enhanced binding affinity to target. Mean you can, by modification, enhance the affinity of the siRNA to bind to target RNA. So the challenge is, first you need to identify appropriate gene targets. You have to know which gene you want to target to get the effect. And the next step, you have to choose or to generate the antisense oligos specific for the gene of interest. The next advantage, or the next challenge is need of to do some modification to make the RNA more stable and to protect from enzyme degradation. The last challenge is drug need to be delivered to the organ of interest, as Dr. Ahmed shows before. So after using or after injection, the drug, most of drug, they will go in the liver or in the kidney. So to get the antisense oligos to the specific targets, this is one of the most interesting challenge in the using of antisense oligos. So to the challenge of modification or the stability of the antisense oligos, there is a lot of modification. Here we can see the first one, or the first normal one, that is phosphodiester, which we found in RNA. The first modification or the first generation of antisense oligos, the modification is in phosphothiazide. Okay? And this one is the most used antisense oligo in the antisense oligo strategies. The next modification is methylphosphonate, which they add here a methyl group and the phosphate group. And the third is for phosphoamidate. You make a change in the phosphor group all of these antisense oligos. This is the first generation of antisense oligos. So few of the phosphatidyl, this is the first generation, reached already the clinical trial in some therapies. So an example for these antisense oligo which are used already in the therapy is the fomavacin, which was approved and marketed for the intraocular treatment for cytomegalovirus retinitis. So RNase H dependent phosphate oligonucleotide produce a vast array of non-specific effect when we use the first generation of antisense oligos. So in the second generation, they change the second carbon in the ribose. They add 2' O-methyl in the first group and 2' fluoro in the second. This both structure is part of the second generation of antisense. Another construct is the locked nucleic acid. They make a bridge between C4 and O2 using methyl group. This structure is a third generation antisense oligos. So normally, drug developed from second generation antisense oligos contain 20 nucleotides, and most of them are 10 deoxynucleotides, which are flanked by 5 ribonucleotides. The third generation antisense oligos, they make complete change in the ribose. So they replace here morpholino oligomers instead of ribose. So these constructs have high affinity and less non-specific property because there is not charge, and they have also high stability compared to the second generation. Disadvantage of morpholino oligos is they can induce p53 apoptosis, and apoptosis can get all the cells, of course, and this is one of the disadvantages of the morpholino oligomers. The second structure is peptide nucleic acid. This is repeating to an amino acyl which have neutral charge and can have high binding affinity. Disadvantage of the peptide nucleic acid is poor cellular penetration. So it is difficult to get it in the cell because it's neutral. So the last one is the locked nucleic acid. In the locked nucleic acid, we found still the ribose construct is not changed, in contrast to the other one, which is completely changed. And then here, they just make lock between C4 and O2 using methyl binding. This is favorable for high binding affinity and the lack of unspecific toxicity. Disadvantage is a high potential for hepatotoxicity. So the summary for the antisense oligonucleotide generation. First generation has moderate ability to penetrate the cell. Stability in vivo is high. Lower binding capacity, higher effectiveness, more pronounced non-specific effect. So the second generation, they have the ability to penetrate cell is moderate. Oh, sorry. High nuclear stability, high binding capacity, high effectiveness, and more pronounced non-specific effect. Third generation, they have high penetration, nuclear resistance, so the degradation is very stable. Much higher binding capacity, much higher effectiveness, and moderate non-target effect. At the moment, the LNA, that is the locked nucleic acid, are currently the most investigated product in the clinical trials. Delivery of antisense oligos, as we mentioned before, is the second challenge in the treatment with antisense oligos. So the most of RNA or antisense oligos, they are charged, they are negative, and these make it difficult for them to transport through the membrane, the cell membrane. As you can see here, after injection of antisense oligos, so the most antisense oligos, they go to the kidney and to the liver. This is the most organ which are targeted by antisense oligos. We found some of them in the bone marrow, in the brain, approximately 0 because of the brain barrier. We have some in the heart, in the lung, ovary, spleen, testes, and uterus is very low. So, to deliver the target cells and tissue remain the major challenge for the treatment with antisense oligos. At the moment, there is also a lot of variety how to inject or how to deliver the antisense oligos. First of them, the microinjection. The microinjection is very important if you have, for example, solid tumor, which you can use, and you can see where is it, and you can inject immediately in the place. The second strategy is the use of the nanoparticle. And as Dr. Ahmed mentioned before, the nanoparticle can be bind also to antisense oligos and targets the delivery to specific area. There is cationic lipid, antibodies, cholesterol, aptamers, and the last one is viral vector. So you can also use antisense oligos for delivery in viral vectors. We pass to the next part, is the mark or the brand name which already in the production or in the therapy. The first one is fomaversin, was first generation antisense oligonucleotide with phosphorothioate that used in the treatment of cytomegalovirus retinitis. Cytomegalovirus retinitis is a virus that affects the retina and then causing inflammation in the AIDS patient. Cytomegalovirus retinitis cause inflammation and lead to blindness in the AIDS patient. Fomaversin bind to major immediate iliary region, that is the target gene or the target region, that is IE, and then cause the degradation which inhibit the production of the cytomegalovirus. It was developed by Ionis Pharmaceutical and licensed to Novartis. In August 1998, fomaversin was the first antisense oligonucleotide which was approved by Food and Drug Association as treatment for the cytomegalovirus. It was used as intraocular injection. Novartis stopped marketing in the EU in 2002 and in the USA 2006 because the treatment with therapy, different therapy for the HIV patient reduced the production of HIV, and this why the need for fomaversin was low, and this why the company stopped the production. The second treatment is mipomycin. Mipomycin is treatment for familial hypercholesterolemia. Hypercholesterolemia is a disease for person what they have they don't have low LDLR, that is low density lipid receptor. A lot of people they don't have the receptor. Is a genetic disorder characterized by high level of low density lipoprotein. The prevalence is about 1 person in 200 to 500 person. And the treatment or the target is the ApoB100. The ApoB100 is a gene which is very important for the production and secretion of the very low lipid density from the liver. So after the treatment of ApoB100, how it happened? So the mipomycin, this is an antisense oligos, which bind to the ApoB100 mRNA and cause the degradation. So after the degradation of the ApoB100, so the secretion of hepatic VLDL will reduce it, which conducted to reduction of the LDL in the body. So different studies showed that the LDL cholesterol levels was reduced by 54% in the patient with LDLR. Canimorab produced by ISIS, which is under Sanofi. January 2013 was approved by FDA as treatment for familial hypercholesterolemia. And low familial hypercholesterolemia, the most people, they have to make electrophoresis. Yeah, this is very expensive, and a lot of the people, they don't have the opportunity. And a lot of people, they die within 30 years. So the treatment with the Canimorab will improve the life of the most of the people. So the next disease is Duchenne muscular dystrophy. Muscular dystrophy is also an inherited disease. So the most of the infected person, they are male, and the incidence is 1 in 3,500 to 5,500 person. So the disease is X-linked recessive. Recessive mean both gene must be affected. Yeah. Here we have, for example, one gene is affected and one gene is normal. So the children, only the person, they have one copy because this is X-linked. So the son will get the disease. I cannot see here. So for the female, they will not get the disease because the X, they will get it from the father. So the most of the female, they will not get the disease, but only the male, they will go to the disease. However, 1/3 of the person, they have DMD, is also new generation. It's not only getting from the mother, but only there is new mutation which cause DMD. And DMD, the lack of dystrophin, cause muscle weakness, respiratory or cardiac complication, resulting in early death. So the most people, they die within 20 years. How it works? The strategy. As we know, the gene, they have exon and they have intron. So the exon, they have the information, but the intron, they don't have the information. After pre-mRNA, the splicing will cut all the intron and splice the exon together, which produce at the end the dystrophin protein. In the affected person, they have mutation in the exon 49 and 50, and the exon 51 is also defect, which after splicing we have 48 and 51 and 52, which doesn't produce the protein. So the principle for the eteplircine is a morpholino oligomer with 30 nucleotide long. It's bind to the exon 51. After the binding and the splicing enzyme will produce exon 48 and exon 52 product, which is shorter, but it will produce an effective and functional dystrophin protein. So the produce is produced by Sarepta Therapeutics. and is approved by FDA in September 2016 as treatment for the AMD. The Huntington disease is also same, is a disease which affect the children, but this disease is dominant and autosomal. Autosomal and dominant mean the children, male and female, they can get it, and the parent both they can transfer also the disease. The problem of the disease is dominant. What's mean? If only one gene is defect, so the children they will get the disease. Okay, no more longer. So, what will happen in this case? We have specific primer which bind to the specific mutation. And the mutation or the copy with the mutation will be degraded. In this case, then we have the functional one which will be free. So, then we have the elevation of the disease using this therapy. Miraversin is also on the same strategy. They use oligomers to degrade MIR 122. That is very important in protein for the replication of hepatitis C. And here there is different application or different drug which under clinical phase 2 and 3 for the treatment of carcinoma, different carcinoma, hepatocellular carcinoma, neuroendocrine tumors, adrenocortin carcinoma, lymphoma, for pancreatic cancer, for ocular hypotension, and for acute kidney injury disease. As other treatment is for lung cancer, colorectal carcinoma, and others, we have the treatment for different inherited disease. We have for asthma, for viral infection, and also for insect and pest management in agriculture and forestry. So the last is show the most important companies. 1980 is the antivirals which changed the name in Sarepta Therapeutics, and ISIS Pharmaceutical, which changed the name 2015 Ionis Pharmaceutical. And there is different companies in different Europe countries which are in production of the antisense oligonucleotide as treatment. Thanks for your attention, and I'm happy to get your question. I'm started late.
Okay. Thank you, Dr. Abdulaziz Bauzi. The talk is now open for questions. As usual, we will start from the female side. Is there any question, please? Yeah, please adhere to question and not to the comments, etc. So we have not much time here. Question, please, from this side. One question. Okay.
I can hear, but this, the audience, they cannot hear.
Hello.
Assalamu alaikum. Sorry for questioning again, but I'm very interested in your topic, doctor, and thank you for this nice presentation and valuable information. I just wonder if we can make use of this antisense mechanism in treatment of the other neurodegenerative disorders, and in this case, we can use the third generation part, and we can avoid the hepatic toxicity by giving this treatment intrathecally. What do you think about this? Thank you very much.
Yes, of course, you can use the antisense oligonucleotide for different diseases, for other protein, of course. The most important, you have to define the gene, the target gene, which gene you want to target, and to define also which oligosense have the effect. Not every antisense oligonucleotide they have effect, but you have also to choose the specific one which can get effect. So there is different program now in the software. They choose different poles of antisense oligonucleotide, and then you have to test it until you get the best antisense oligonucleotide for the use. As strategy for the locked nucleotide acid, this is the most powerful one The disadvantage, of course, is the hepatotoxicity, and there is different strategy to reduce the toxicity of the hepatitis. For example, the use of antibodies or nanoparticle coupled to antibodies to make the targeting more specific. This is one. Or you have to adjust the doses to get low side effect.
Okay, thank you. Okay. What about the male side questions? You give the mic to sir here.
Thank you very much again. I think it's very interesting. Very interesting topic. You can hear me, right?
Yeah, I can hear you.
Everybody? It's fine. So regarding the clinical applications, there are already some medications available in the market. For SMA, for Docini, now nusinersen is already in practice. In Saudi Arabia, we have 200 patients already have Docini or SMA, spinal muscular atrophy. These medications also come with a significant cost. They cost millions of riyals for the management, for example, of using Spinraza, which is nusinersen.
Yes.
For SMA, it's costing 4 or 5 millions. The tricky part is the selection of patients also who will benefit from the medication. They have to have a certain genetic expressions. Also, it has to be used early on in life before the damage take place, especially in neurological diseases like SMA. This is applicable for so many other gene therapies. The cost, the way of delivery, it has to be given intrathecal, for example. Yeah. It has to be done through under imaging guided. It is not an easy administration. And the cost is continue to be very prohibiting from a practice point of view. Do you see in the future, in the development of the technologies, the cost of these new inventions in therapy will be coming down somehow?
Yeah, of course. Everything, it starts at the beginning is very expensive. When you want to buy the LCD TV at the beginning for 5 years or 6 years, the price was more than 40,000, 60,000. You know, you can get it for maybe 10,000 or 4,000. Everything, when it started at the beginning, is expensive because the research takes more time. It takes more than 20 years from the funding for the first step when you found the effect until to get the license for marketing is take more than 20 years, and these make the cost very high. And all the companies, they need to get their cost back as soon as possible. Yeah. I'm sure that the cost will be lower. And of course, if there is also other system, like an insurance system, which encourage or help and support the people to get the disease, to get the treatment, even if the cost is expensive, this will encourage the production and the development of these treatments. So I think this is very important. We have to take it because a lot of people, they have most inherited disease. They tell them you have to wait and wait today. That's it. So you cannot get anything. And then for one person who wanna live longer, he must take the treatment. How much is it?
And also for this therapy, you have to use it early in life. So most of SMA, they have been used within 3-6 months of life. Otherwise, the neurological damage already taken place, and it's a waste of money and time to give these medications.
Yes, this is true. Yes, I agree with this. The therapy have to start, but there is also other therapies. They start in the late stage, and then they found there is improvement also. Yeah. But if there is opportunity to take the treatment from the beginning, is much better.
Thank you, Dr. Abdul Latif. Is any question this side? Yes, please. Give the mic to him.
Get mic.
Abdullah, can you help with the mic?
Assalamu alaikum.
Alaikum salam.
I noticed that fomiperazine was withdrawn due to its hepatotoxicity. Also,
I don't hear it.
Fomiperazine was withdrawn from market due to its hepatotoxicity. Also, mibomersen have a black box warning from FDA due to its hepatotoxicity. There is a link between the mechanism.
I don't hear the last question.
There is a link between hepatotoxicity and the mechanism of action.
What is this?
Fomimersen and mepomersen both produce hepatotoxicity. Yeah.
Yes.
There is a link between the mechanism and hepatotoxicity.
No, I think, but there is sometimes there is maybe there is in specific binding to some genes which are important for the life of the cells, and because of the binding, because the binding is also very high, it's cause the destruction of the mRNA, which is cause the apoptosis of the cells. That is, most of them, these is unspecific binding. But until now, I don't think they have the mechanism. They don't identify the mechanism according to the hepatotic toxicity.
Okay, is there any more question?
Assalamu alaikum.
Alaikum salam.
You are mentioned for more disease. You mentioned the male. How to know the male more disease and effect? And you talk in the X-linked have more than Y-linked for female.
No, the X-linked, especially if the disease is recessive, yeah. When the disease is recessive, that's mean both copy must be affected, because each one of us have two copy. One copy is from the mother and one is from the father, okay? And the recessive genes, they must both defect to get the disease, okay? When the male gets X from the female, which have the defect. How to know? Yeah, because there is disease, you know how is inherited. This is based on early study. For example, give you an example about hemophilia. Hemophilia is known since long times that is X-linked recessive. That is principle. If you know this is X-linked recessive, then you know immediately who will get the disease.
Thank you, Mark.
Welcome.
Okay, thank you, Dr. Abdul Latif. It was a nice and interesting talk about the antisense oligonucleotide and approach to the genetic diseases. Ladies and gentlemen, please be seated here, and we will start the opening ceremony followed by the poster session. Thank you for your time and attention for the first session of the symposia. Thank you again. Thank you very much.